Daniel's already given a good intro, where we're now moving from trying to find a target to trying to screen drugs that can impact that target. With that,
Okay,
Mojtaba, the floor
uh,
is yours.
thanks, Daniel. Thanks, Andrew. Okay, perfect. Hello, everyone, and thank you for joining us today. The topic of my talk for today is how we can find new drugs for a parasitic disease. Okay, so this is Joe. Now that you see him, he is asleep. He suffers from human African trypanosomiasis or sleeping sickness. It is definitely different than the common insomnia that people get every now and then, and it is fatal if left untreated. He was bitten by a tsetse fly that you see its picture here, and upon which these single-cell parasites were injected into his blood. These parasites are called Trypanosoma brucei. A scientist named David Bruce found them, and these parasites were named after him. They endanger half a billion people, and we need new drugs to help Joe and other people. So let's see what we can do to help them. There are two different approaches in drug discovery. The first approach is phenotypic screening, in which we try to find a compound that kills the parasite in the first place. And then in the next step, we will try to define how this compound is actually killing the parasite, and then we will optimize it. In the second newer approach, which is called target-based screening, we have an essential target that is unique in the parasite, and then we will try to stop it, and then we will determine if the compound can kill the parasite. As you may remember from Daniel's presentation, his research was focused on the first part of this approach. My research mainly focuses on the high throughput screening and finding the compound that can stop this process and the further tests that needs to be done. And it is highlighted here. Okay. So in the first step, we need to have an essential target inside the parasite, which is absent in the host, which is us, the humans. In this case, it is kinetoplast, which is simply the mitochondrial genome of the parasite. So let's take a closer look at it. As you may remember from central dogma theory, in cells, DNA gets transcribed into mRNA, and then the mRNA will be translated into protein that has many different functions. But something strange happens in these parasites. The mRNA that got transcribed from the mitochondrial genome or the kinetoplast needs to get edited in order to become ready for translation. And this addition entails uridine insertion or deletion. For example, here we see an mRNA that upon addition, it gets two more U's in its sequence. But how does this happen? A protein complex called editosome is responsible for this addition, and it is essential for the parasite's viability. So if we find a compound that can stop this protein complex, this addition will not occur, and the parasite will die. Now that we have identified our target, which is RNA editing and editosome, we have to find the compound through high throughput screening. And what I'm going to show you here is the workflow of the high throughput screening. In the first step, we will screen 1 million compounds, and among them, a few thousand compounds can inhibit the RNA editing. And then we refine them, we will filter them and measure their potency, their efficacy, and see if they can kill the parasite. Then we will have some candidates, and then we need to test them in preclinical development stages in mice and humans through clinical trial. Okay, so this is the first step. The first step is the big screen. Here we tested about 1 million compounds, and this is the result of the high throughput screening. Each dot represents a compound, and the x-axis shows the compound number, and the y-axis shows the percentage of inhibition, which means to what extent a compound can inhibit the RNA editing process. The red line is our threshold, so if a compound lies beyond the line, it is considered as a hit or inhibitor. So now that we have performed the high throughput screening and we have 1,000 hits, we will refine and filter them and select the best to have 100 compounds for further tests. Then we have to test their analogs, which means we will have to test the compounds that are similar to the hits that we found. And then we will test if they can kill the parasite and if we can make them more efficient. Also, we have some assays that can help us understand if the compound is actually inhibiting a specific step of RNA editing so that we can make it more efficient. Okay, after performing all of these assays, we will have 1 to 3 different candidates, and further experiments include testing them in mice and then the humans in clinical trials. So here, these are 2 different gels that we will try to define the mode of action of the drugs against the RNA editing. So for example, here you can see this mRNA, which is this band on the gel. And if the editing happens, we see the shift, the 4 different bands, which means these U's will get deleted. And if we have an inhibitor, we see no shift, so there is no editing. So these compounds might serve as drugs. And the same things happens here where 2 different RNA substrates gets ligated together after deletion of these U's. So at first, these U's will get deleted and we see 4 different bands. And these bands will ligate to another RNA substrate that we see here. So if we have no inhibitor, we should see it like this. But if we have an inhibitor, that actually a compound that can stop the RNA editing, we see it like this and no shifts. Overall, after performing all of this research, we will have some lead compounds that can be used as drugs for the human African trypanosomiasis. And if you're still not satisfied, these drugs can also be used for similar parasites that can cause many deadly diseases. And with that, I want to thank my supervisor and my lab members.